using sterile pipette with 7 mm in diameter, we put the propagule and then dip it into the CC solution. We wait for 30 minutes until the bit formed and then wash with sterile aqueous for 3 times. The bit stored in the plastic dish sterile with wet filter paper inside. As the first factor, we use combination of sodium alginate and calcium chloride. There are 3 concentrations of sodium alginate. Those are 2, 3, and 4% of sodium alginate, and then 75 millimolar and 100 millimolar calcium chloride. As a second factor, storing at temperature 4 degrees Celsius in refrigerator with no light, with interval time 1 until 8 weeks. For germination testing, the beets planted in the germination medium. In this study, we use hyponic modified medium, and then culture stored in room temperature with periodicity 16 hours light per day. And germination and growth process are observed for 3 months. For parameter observed are germination percentage, time to produce shoot, plant height, number of leaves, and shoot. For data analysis, we use descriptive analysis for artificial seed characters, and for germination percentage, time to produce shoot, plant height, number of leaves, and shoot, we use ANOVA test and continue with Duncan multiple range test. And the result for artificial seed characters, for 2% of SA, the bead is not fully rounded, not definite in shape, with a tail in one side of the seed, and also too soft to be handled. This concentration is not suitable for artificial seed formation. 3 and 4% of SA gave uniform isodiametric and solid shape of the seed, but the difference between those 2 concentrations was the size of the seed. The 4% SA will produce bigger diameter seed than 3% of SA. The seed with 75 millimolar CC will give clearer surface, as you can see in figure A, C, and E. Thus, the protocoms inside the encapsulation matrix can be seen clearly. Meanwhile, the higher CC, as you can see in figure B, D, and F, will give the milky white matrix and rigid wall. From this result, 4% of SA and 75 millimolar of CC is the best combination in producing qualified artificial seed of Dendrobium macrophyllum variegatans. In Table 1, the storage interval at 4 degrees Celsius affected the germination rate, and 1 week storage gave the highest germination rate. In this study, 91%. The seed had high viability up to 4 week storage and still can germinate well until 8 weeks. And from our observation, the seed will lose its ability after 8 weeks, and the symptoms shown that the protocom being brown. And from Table 2, we can see that combination of sodium alginate and calcium chloride, and also the storage interval after storage gave the effect of germination time. And we can conclude that the longer of the seed stored will need more time to germinate. In Table 3, we can see that storage interval week gave the significant effect on the plant height, leaf formation time, and number of leaves. And the 5 week storage gave the highest plant height, most number of the fastest leaf formation time, and the most number of leaves. And in Table 4, only combination of sodium alginate and calcium chloride gave the significant effect on the number of shoots, and we can see 4% of SA and 75 millimolar of CC gave the most number of shoots. This is the example, the plantlet with from the seed with SA 4% and CC 75 millimolar, and the seed with SA 2% and CC 75 millimolar. The seed with SA 4% and CC 75 millimolar gave the most number of shoots. As the conclusion, in this study, the isogeometric, uniform, and solid shape of artificial seed of Dendrobium macrophyllum alternatans can be produced by using 4% of sodium alginate as matrix encapsulation and 75 millimolar of calcium chloride as gelling agent. The encapsulation technology gave promising result in interim storage at low temperature that the artificial seed of the species can be stored for 8 weeks in low temperature. In the study, 4 degrees Celsius, and the 5-week storage interval of artificial seed of Dendrobium macrophyllum alternatans gave the higher germination rate, the highest plant height, the fastest leaf formation time, and the most leaves number. Thank you for your attention.
Thank you for the nice presentation from Bu Poppy. I would like to clarify first before we show the next video. Is Bu Santi Ratnakomala here in our break room? If not, then we need to move to the next presenter first. And hopefully Bu Santi will join us for the last video presentation as the 6th video presenter. So we will move to the next presenter here if there is no Bu Santi here. The next one will be from Bu Sri Lestari. The presentation will be about the role of Michelia champaka agroforestry system in combating global warming. So please.
Assalamualaikum warahmatullahi wabarakatuh. It is my great pleasure to be here to present my paper in ISIBio 2022. Let me share my presentation first. Today I would like to present the role of Michelia champaka agroforestry systems in combating global warming and providing livelihood options. This paper is written by me, Sri Lestari, and Bondan Winarno. We are from Research Center for Ecology and Ethnobiology, National Research and Innovation Agency of Indonesia. This is the outline of my presentation. The first one is introduction, the second one is method, the third one results and discussion, and the last one is conclusion. Indonesia being a major emitter of greenhouse gases along with the increase of CO2 emissions caused by forest and land degradation both in the uplands and lowlands area. Illegal logging, forest and land conversion, agricultural crop development, and expansion of settlements are factors influencing forest and land degradation. The environmental damage that continues to occur throughout all regions make them more vulnerable to the effect of global warming. Coupled with the implementation of land management systems that pay less attention to sustainability aspects, results in the declining condition and quality of land and disruptions of the ecosystem on Earth. In the long term, it will cause harms to society and the environment, reduce production and reduce environmental quality, which is in turn has an impact on the health and humans and other living creatures. The agroforestry system has been promoted as one of sustainable land use management, which is expected to improve environmental conditions. Agroforestry as a promising land management option can affect households' perception about the seasons around them. Agroforestry is a safety net for the family economy when there are fluctuations in price levels and the number of production on agricultural products. So that this paper aims to investigate the role of agroforestry system of Michelia champaka in South Sumatra in combating global warming as well as protecting natural resources and providing livelihood options for the community. This is the method. research location focused on Ogan Komering Ulu Regency, South Sumatra Province. This regency was chosen because some of the community who live in these areas has developed Miselia champaka agroforestry for several years and gained benefit from this species, both tangible and intangible value. These three species has been widely developed, either monoculture or mixed with other types of plants such as coffee, fruits, and cocoa. For the data collection and analysis, observation methods and interviews were carried out to obtain the data. Field observations were conducted in the community garden where Miselia champaka has developed. This research activity was conducted in 2018 and 2019. The data collected includes the reasons why farmers choose Miselia champaka to be developed on their area, how the planting is carried out by the farmers, as well as the financial benefits obtained by farmers from developing this Miselia champaka. And finally, descriptive qualitative research analysis was used as a data analysis tool. Data and information were analyzed descriptively to get an overview of how Miselia champaka developed by the community in agroforestry system plays a role in combating global warming and at the same time becomes a source of livelihood for the community. This is the result and discussion. Agroforestry system of Miselia champaka and livelihood options. Miselia champaka is widely developed in some regencies in South Sumatra province, including Empat Lawang, Lahad, Pagar Alam, Ogan Komering Ulu Selatan, and also Ogan Komering Ulu Regency. The dominant system that has developed by the community was mixed pattern that is known as agroforestry system that has been practicing for generations. Initially, utilization of Miselia champaka was for subsistence of household construction. Following the timber supply for the forest was continually decreasing, this timber was one of the options to supply the market timber. Therefore, this timber tree gained a significant increase of the market price and had become the prospective timber commodities for commercial purpose at regional southern Sumatra. The main crops grown by the community in the research location were coffee, rubber, cacao, and maize. Apart from planting Miselia champaka, they also grow fruit trees. You can see here from the picture. Based on a financial study on Miselia champaka with coffee crops in South Sumatra, it will give at an interest rate of 13%. This pattern can produce an NPV up to $2,371 with a BCR of 2.33 and an ERR of 38%. Miselia champaka can be harvested and produce good quality wood after it is over 10 years old, and these three species support the livelihoods of the community from the timber and seedling production from the fruit. Scoping Miselia champaka agroforestry for climate change. Most of the population in low-income countries, including Indonesia, relies on agriculture for their livelihoods and rural development. However, they are more vulnerable to climate change because they have limited ability to invest in more adaptable institutions and technologies. Agroforestry is an alternative option to intensify sustainable agricultural practices to improve food security by using effective management techniques, both socially and cost-effectively. Agroforestry provides ecosystem services such as regulation of the hydrological cycle, prevention of soil erosion, carbon sequestration, and the most important is the production of food and other supporting products for the community. Raising awareness of the community to the role of timber trees, such as Miselia champaka for ecosystem services, is less attention by stakeholders at the field level. Miselia champaka agroforestry in combating global warming was observed qualitatively in the research area by interviews and discussion with the farmers. Some of them mention that the Muselia Champaka tree in the middle of his land has the effect on reducing the air temperature. The wind is still blowing slowly and the birds are starting to arrive, even though some of them haven't stopped, maybe because the tree is not big enough. Some of them mentioned that the growth of Muselia Champaka in my lands was good. Therefore, in the future, I will replace the coffee seeds trees with Muselia Champaka, and it will be more beneficial and support environment conditions. Maintaining land use with agroforestry systems allow above ground carbon stock up to 50 megagram per hectare and minimizes biodiversity loss from the land. This is the study result from Sari et al. 2020. For the conclusion, agroforestry techniques are important for the purpose of restoring and rising land productivity. Agroforestry is often considered a cost-effective climate change mitigation strategy because the tree-based management systems that are widely developed by the community are able to store carbon in soil, woody biomass, and other types of crops that are simultaneously grown. The impact of climate change is often not directly realized by the community, but the impact has been felt by the community in their daily lives, including change in temperature and seasons. Agroforestry has significant impacts in increasing soil fertility and improving local climatic conditions, as well as providing ecosystem services that benefits the community. Thank you very much. That's all of my presentation. I will be happy if I get some input from you all. Thank you very much, and wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. The next presenter is the most handsome presenter in our session today, which is Pak Said Fahmi. Will present about the food plan, biodiversity, and conservation of upland landscape in Indonesia for sustainable development. Please, Pak Imam, turn on the presentation video from Pak Said. Thank you.
Assalamualaikum warahmatullahi wabarakatuh. Hello everyone. My name is Said Fahmi. In this paper, we have 5 authors, including me, Bondan Winarno, Lutfan Nugraha, Sri Lestari, and Tri Wirayuwati. We are from National Research and Innovation Agency, BRIN. Today, I would like to present my paper about food plant biodiversity and conservation of upland landscape in Indonesia for sustainable development. So, introduction. In the last few decades, the world's population growth has been significantly fast, including Indonesia. The rate of human population growth has an impact on food availability. Conversion of highland into industrial agriculture often occurs in several districts in Indonesia. However, food security is still an important issue that has not been resolved. In fact, industrial agricultural activity is one of the causes of the land degradation and loss of biodiversity. Harmonizing biodiversity conservation with the development has been a challenging issue for sustainable development in Indonesia. The government has made policies related to food security as land use. As we can see in the slide, the government has implemented PHBM managed by Perhutani for sustainable development to increase food production. For example, of the application of PHBM in the protected forest of South Bandung, with an agroforestry pattern between eucalyptus and coffee plants. The application of the agroforestry is able to overcome forest damage and forest disturbance. But this system creates discontent among farmers. then they are destroying the agreement, and it's make a conflict. So in this paper, we will take up how the implementation of upland use systems applied by community and sustainable upland farming used for landscape conservation and food security. Research scope, there is 3 issues that we review. That was agricultural land crisis, food plan sustainable upland farming, and food security. From these 3 issues, the question is, how is the relationship between agriculture productivity, food plan biodiversity conservation, and sustainable landscape management? This paper will examine the biodiversity and conservation of food plan for food security towards sustainable development in the upland of Indonesia, and to find out what factors influence of food plan biodiversity and landscape conservation. Methodology. In this research, we use a qualitative descriptive method, which is divided into 3 stages. First, data collection. There is observation and triangulation. The second one is the determination of data collection. There is document analyzing, reviewing, and sanitizing. And the third one, data analysis technique to reduction data, presentation data, and verification. The study was limited to the paper which was published from 2007 and to 2022. Result and discussion. There is 3 content. The first one, biodiversity of food plant species in the upland Indonesia. Second one, upland use system applied by community. And the third one, towards sustainable upland use for the conservation and food security. Based on the result, we share the types of food crops in Indonesia. In this slide shows the dominant types of food crops are grown in the upland and also type of agrosystem. We are divide the agrosystem by 3 into farmland, home garden, and garden. Sumatra Island shows the majority of food crops are Oryza sativa, with the different local name in each regions, and the type of agrosystem that often used is farmland. In Heram district Japapura, areas is to be vegetable, fruits, and tubers, most majority of food crops. And Java Island, the food crops grown are more homogeneous. The type of agrosystem that is often found is home garden. So to the second one, upland use system applied by community, there is 2 provinces that we want to discuss. The first one is North Raja regions. In this province, the application of an agroforestry system on coffee plants with shading trees is potential way to improve the agricultural system and sustain the livelihood of the local community upland areas, while serving to protect biodiversity. Agroforestry between Arabica coffee with Kaliandra show the optimum productivity of coffee in North Raja, South Sulawesi. In the other hand, Kupang district also have agroforestry in the upland farmers. They continue to use conventional or traditional farming methods and collect honeybees. The majority of upland farmers engage in shifting cultivation with slash and burn farming with little to no external inputs. The agroforestry system can cover food security issues. It has started to be distracted by the influence of the economic pressure. Monoculture or intensification counts as one of the capitalization side effect that could not be avoided because of the embedded market-based approach grand idea. This situation has done nothing better for food system but only increasing certain kind of food availability more than before. Last but not least, toward sustainable upland use for conservation of food security. This is the important thing to reach towards sustainable upland use upland use for conservation and food security. First one, about land management. The main problem of land management in the upland area is mainly soil degradation due to erosion. The most important factors that affect the yield of erosion are the slope of the land and the type of the plant factors. Land management practices carried out by the surrounding community often pay less attention to the sustainability of land resource because they prioritize agricultural production as their source of income. The second one, about maximizing agricultural efficiency. There are three factors that affect biodiversity as well as food security. First, modernization and sustainable agriculture. Second, knowledge and management. And the third, governance and process. And the third one, policy and guarantee. The government, along with the other stakeholders, should conduct socialization to the community, educate the community, and provide an example of good, profitable upland management system for the community. Guaranteeing market availability for planted products is also very important because the market aspect greatly affects the willingness of the community to plant certain types of plants. Conclusion, the complex socio-ecological system and environmental change in Indonesia upland area have occasionally put in the local population and the environment at risk. In upland area, poor land management methods harm ecosystem and soil, threaten biodiversity, and fail to take into account conservation issues. The majority of crops cultivated in the uplands are agricultural crops such as Oryza sativa, jenis, Comea batata. The third one, upland farming should be improved by integrating technological, institutional, and social engineering. Agroforestry and other agricultural focused livelihood initiatives are essential but not sufficient for attaining conservation objective or protecting human life. That's all from me. Thank you for the attention. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Pak Said, for the presentation. For the last time, I would like to confirm, is Bu Santi Ratnakomala here in our break room, or the representation of Bu Santi is here in the break room? If there is no representation of Bu Santi or Bu Santi itself, I think the presentation of the video is finished. And now we can move to the Q&A session. We will have approximately 20 minutes for the session until a quarter hour before 3 p.m. We already have several questions from 3 participants here in the parallel room. The first one is from Bu Tri for Bu Retno. We have 2 questions here. Please, Bu Retno can answer and confirm the question. The first one is, why is the density of soil microorganism in the polyculture area was lower compared to the monoculture area? And the second one is, are these plantations located on ex-nickel mining area? If so, will it give impact to the soil microorganism abundance? So please, Bu Retno, time is yours.
Thank you, Pak Deki. Thank you, Bu Ira, for the question. I will try to answer your question. The first one, why is the density of soil microorganism? Yeah, actually, we have when we collected the soil samples, we interview the owner of the land or the pepper plantation, because when we collected the soil sample, we also saw the growth perform paper plan in the polyculture cropping pattern, the plant have what's called growth response not good. Then the pepper plant in the monoculture cropping pattern. So we interview to the owner, and we ask what they use or what they apply for their plantation, and they said that they use chemical fertilizer in the polyculture cropping pattern, but in the monoculture cropping pattern, they use organic fertilizer like compost. So maybe that's why the density or the abundance of soil microorganism in the polyculture pattern is lower than monoculture cropping pattern. Besides that, we also I will answer your second question. Yes, you're right that the location of a pepper plantation in the nickel X mining. So that's why the population of the soil microbe is lower, and we also know in the nickel X mining contain high of heavy metal like nickel, chrome, cadmium, and so on. So maybe that's why the population or the density of soil microbe in the area is lower. Maybe that's all my question. I hope can answer Bu Wira question. Thank you.
I think if I can summarize that, the factor that causing the differences of soil microorganism is the application of the synthetic fertilizer in the polyculture. And the second one, yes, it is on the X.
Yeah. Yeah.
Hopefully it is sufficient answer for Bu Wira. For the next question is from Bu Poppy for Bu Alfia. So this is very technical about tissue culture method. It also stated in the chat menu. I will mention it directly now. For Bu Alfia, are there any necrosis symptoms during the application of PBZ, paclobutrazole, to the explant? Since the dose of PBZ is quite high, and I found the symptom in the orchid culture as well. And the second one is how long the abnormal or retard plant become normal again? Thank you. Oh, please, time is yours, Bu Alfia.
Okay, thank you. Is my voice clear? Hello?
Yes, but you may want to increase it if you want. But it is audible. Please.
Oh, okay. Thank you, Mbak Poppy, for your question. I try to answer it. In sugarcane conservation research, using paclobutrazole with doses 2, 4, and 6 milligrams per liter, we found no necrosis symptoms occurred to the explant. Maybe because every species has different responses to paclobutrazole. Even in sugarcane itself, some variety show response to paclobutrazole, but the remaining variety not show a single symptom in morphology. To the second question, how long the retard plant become normal again? The retard plant became normal again after 3 cycles of subculture, with interval between subculture is 3 weeks. Hope you satisfied with my answer.
Okay, thank you. So, Bu Poppy, I would like to clarify. I think Bu Alfia said that the PBZ is not giving that kind of response to the explant of sugarcane, I assume, and the return plan become normal again in the period under 1 month. Is it right? 3 weeks or 4
Yes,
weeks?
3 weeks. 3 weeks.
So it's under 1 month. Hopefully that answer your question, Bu Poppy. Because we have more question here, at least 2 questions, I would like to move to the next one. Bu Poppy, if you have want to have more discussion, you may want to continue it after this. From Bu Safina to Pak Said. Can you explain what indicators you choose in determining the upland area in that paper? So what is the indicator you use here? And then the second one is why monoculture and agriculture intensification are not recommended in the Indonesian upland area. So please, Pak Said, time is yours.
Yes, thank you for Mr. Moderator, Bapak Diki. Is it my voice is clear?
Very clear, Pak Said. Lajengkeun.
Okay. Yeah, the indicator that we choose to determine the upland area is we follow the regulation of the Ministry of Forestry and Environment, number 33, that about guideline for preparation of climate change. And in that regulation, that definition of the upland area is above 700 mdpl or MASL. So all the paper that we review, the location area is above 700 MASL. I hope the second one. Sorry, the second one.
Yes. Why monoculture and agricultural intensification are not recommended in the Indonesian upland area? Why it's not recommended?
I'm trying to answer based on our review. Based on our review, that find out the most erosion occurs when the horticultural crops or seasonal crops are grown in monoculture is over dangerous than the agroforestry system. Can I have 1 minute to give an example? That I have a paper from Adriani et al. That find out in the tobacco upland area is erosion is very high. It's about 531 milligram hectare per year. And then if you want to compare with agroforestry system and monoculture coffee system, the erosion with agroforestry system around 4 to 7% erosion index, while the runoff with the monoculture coffee system is around 10 to 15% erosion index. So right now in the several upland area is used to the monoculture system that we not recommended to use in the upland area in Indonesia. So if we could to recommend the suitable system that can use to upland area in Indonesia is agroforestry system. Maybe that's all my answer.
Okay, thank you, Pak Said. I would like to confirm the answer again for Bu Safina. So the definition is above 700 for the upland area. And then the monoculture and agriculture intensification is not recommended due to the erosion issues, which is quite significant compared to other methods such as agroforestry. Thank you very much for the answer, Pak Said. And then the last question for this session from Pak Said to Ibu Sri Lestari. Collective actions are needed in combating global warming. How the community on your research area deal with this activity? Please, Bu Sri.
Okay, thank you very much, Pak Moderator, Pak Deki. And yeah, thank you, Pak Said Fahmi, for the questions. I would like to try to answer your questions. Yeah, actually, I do agree with your statement that collective actions are needed for combating global warming, because if only one or two or only a limited number of the community committed to plant the Miscelia champaka, especially in our research area, the impact is not widely enough. But if most of the community try to plant this tree, and by agroforestry system especially, then what can I say? The climate condition can be changed by this activity. And however, when we talk to the community in our research area, when we ask them about how the climate change around like this 10 years later, then they said that especially not really much change, because sometimes the community who live in the village not really understand about what is global warming, what is the climate change, and then they not really understand whether the climate is changed in their area. But when we talk about livelihood options, when we ask them, when we tell them that this kind of tree can help them and can increase their income at the end of the rotations, almost 10 years or more, then they have more eagerness to plant this type of tree. And because most of the community planting agricultural in their area to support their income, then we can ask them to plant this species around in the border area, so not in the middle of their land, because sometimes they're afraid that if they plant trees, then it will reduce the productions of their agricultural crops. That's why we can ask also the the stakeholders in the area that, okay, we have to ask the community to plant agroforestry system or planting trees, so that we can manage our climate conditions at the end of purpose of this agroforestry system. Thank you.
Mantap.
Okay, thank you. Pak Said, is it clear? I think it's also a challenge that the community aware with the global climate change issue itself. So is there any response, or that's enough, Pak Said?
Enough, moderator.
Okay, thank you very much. We still have one question, I think, for this Q&A session. Is there any one more burning question related to the presentation? If there is no other question, I would like to ask one more question to Bu Poppy. Bu Poppy, you present the seed technique for orchid conservation. So regarding the species, so what do you think is it the technique promising, particularly for the endangered orchid species ex situ conservation? What is your response? Thank you.
Thank you for the question, Mas Deki. This artificial seed method is quite effective for, especially for threatening plants, and also for the plant that produce no seed. So we can store the genetic diversity of the plant, especially for endangered plant, and short or mid-term. For long term, we can use the following method by cryopreservation with storage in the liquid nitrogen. That's it, Mas Deki.
Okay, thank you very much, Bu Poppy. And that's conclude our parallel session in our room 6. And thank you very much for all the presenters. Unfortunately, we cannot play one of the video presentation because the presenter is not on our room. Thank you very much for all the nice presentation and also a fruitful discussion and question. And I hope we can get benefit from this session. And I hope it will give you more point of view in terms of the environmental sustainability. After this, everyone please leave this breakout room 6, go back to the main room for the next session of this ISIBio seminar for day 1. Thank you very much. Thank you, Pak Imam, for supporting playing the video. Thank you, everyone. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam
Waalaikumsalam warahmatullahi wabarakatuh.
Terima kasih.
Thank you, Dr. Becky.
Terima
Thank you all the
kasih.
participants.
Thank you.
Please
Thank
can go back to the main room.
Recording in progress.
Welcome back to the main room, ladies and gentlemen. The next agenda is plenary sessions for this afternoon. Therefore, I may invite Dr. Ira Handayani as the moderator of the sessions. But before that, allow me to read short profile of Dr. Ira. Dr. Ira Handayani is currently a researcher in Research Center for Applied Microbiology, National Research and Innovation Agency, the Republic of Indonesia, who are interested in molecular genetics, microbiology, and bioinformatics. She obtained her bachelor degree in chemistry at University of 11 Maret, and she continued her master degree in biochemistry at Bandung Institute of Technology, or ITB, and also her PhD in chemical engineering at Tübingen University, Germany. Dr. Ira, time is yours to lead the sessions.
Okay, thank you, Ms. Aulia. Are you hear my voice? Okay. Hello, are you hear my voice?
Yes, we can hear you
Okay.
clearly.
Okay.
Dr. Ira.
I don't know whether Yvonne already in here or not.
Yes, good afternoon, Ian.
Oh,
I hear you and I'm already here.
Okay, how are you?
Thanks, fine. Hope you too.
Okay. Okay,
Thank you. Okay, I will start the presentation. The plenary session for this seminar. First of all, I'm very happy to see you here and welcome all of you to this plenary session. Before the presentation starts, let me tell you how the presentation will go. First, our first speaker, Professor Yvonne Mas, will be invited to present the material, and then our second speaker, Professor Claudia Tribelli, will also be asked to present his material. After both speakers present their material, there will be a question and answer session followed by a conclusion. Now let's start with our first speaker, Professor Dr. Yvonne Mas. Okay, this is the CV of Professor Dr. Mas. Professor Dr. Mas is a professor of applied microbiology at the Technical University of Brunswick and head of the Department of Bioresources for Bioeconomy and Health Research at the Leibniz Institute, the SMZ, a German collection of microorganisms and cell culture. She has experience as an editor in the journal Molecule and the journal Pointer in Microbiology, and experiment as a member of the topical advisory panel in the Journal of Antibiotics. She is interested in research in the field of microbiology, biotechnology, actinomycetes, antibiotic, and regulation. Now allow me to welcome Professor Dr. Yvonne Mas to make her presentation. Professor Mas, please explain your material. And time is yours, Yvonne.
So thanks, Ira, for this nice introduction, and good afternoon to everybody.
Okay.
And okay, now I see my slides. Okay. Okay. So again, good afternoon to everybody. Yeah, thanks for the nice introduction. Actually, I'm here at the Leibniz Institute DSMC, so the German collection of microorganisms and cell cultures in Braunschweig, Germany. And it's a great honor to give a talk during your session at the International Symposium of Innovative Bioproduction in Indonesia. And today I would like to introduce you into the topic of how we can exploit the biodiversity for novel natural compound production by using regulatory switches. So I should, now it's okay. So before I go into that more in detail, I first want to mention why this kind of research is relevant at all. And I think all of you know that antibiotic research indeed is relevant because we observe a growing crisis from antibiotic-resistant bacteria. So the World Health Organization regards the antibiotic resistance as one of the 3 biggest threats to global health. So of course, it's urgently needed to find novel antibiotics to combat these drug resistances. But the question is where to find them. I'm sorry, the presentation is a bit slow, but I hope we can do that.
I think it's okay.
Okay. So we can, of course, find antibiotics a lot in actinomycetes. So actinomycetes are known to produce 2/3 of all antibiotic in clinical use today. And this antibiotic production potential is known for quite a long time. However, what is not known for such a long time is that the genetic potential for the production of antibiotics is even larger in these organisms. And this appeared when the first genome sequence of such an antibiotic producer has been sequenced. So this was the genome sequence of the model organism, Streptomyces silicolor. And that silicolor strain is known to produce up to 2 to 4 secondary metabolites under standard lab conditions. However, the genome sequence analysis revealed that the genome harbors up to 20 different biosynthetic gene clusters. However, not all of those gene clusters are expressed, meaning not all of these substances are really produced, but a lot of them occur as so-called silent antibiotic gene clusters. And this is not only true for that organism, but nowadays, since we have a lot of more than hundreds of genome sequences available from different actinomycetes, it turned out that this is true for a lot of these producing organisms. So obviously, actinomycetes still hold a huge potential for the production of novel secondary metabolites. And of course, the current trend is to activate these kind of silent gene clusters. And now I would like to introduce you into the topic how we can activate those kind of silent gene clusters in actinomycetes. Okay, and we actually can do that by using regulators. And regulators indeed are the set screw to unlock the biosynthetic potential of these natural compound producers. However, the problem is just that there are so many different transcriptional regulators abundant in actinomycetes, especially in streptomycetes, and these can be of different classes, and they can interact in quite complex cascades with each other. And depending on genome sizes, those can actually be more than thousands of different transcriptional regulators. So the question is, which is the most suitable and most potent type of regulator that is suitable to be used as a general activator of antibiotic biosynthesis. And today I will focus on these subtype regulators, since we think that these are good candidates to be used as general activators of antibiotic biosynthesis. Okay, presentation is still slow, but okay. So first of all, I will introduce you where we found this type of regulators. And we indeed identified them in the antibiotic producer Streptomyces pristinus pluralis, which is kind of our working horse. So this strain produces the pristinamycin substance, the streptogramin antibiotic pristinamycin. And pristinamycin is actually two different types of antibiotics, the peptide antibiotic pristinamycin 1 and the polyketide antibiotic pristinamycin 2. And the Streptomyces pristinus pluralis strain produces those two substances in a specific ratio. They interact synergistically with each other, and a semi-synthetic derivative of that substance is on the market. It's called Synergid, and it's used as an antibiotic of last resort against highly resistant pathogens. And what we did back then was that we characterized the antibiotic biosynthetic gene cluster. And back then, this was the largest antibiotic gene cluster that has been described so far. And the genes that you can see here in red, they are needed to, they encode for the peptide antibiotic pristinamycin 1, and the blue ones encode for the polyketide antibiotic pristinamycin 2. So when we had a closer look into this antibiotic gene cluster, then we identified a set of 7 different transcriptional regulatory genes, and they belong to different types of regulatory families. And actually, this is quite a large number, because usually these kind of antibiotic gene clusters have 1 or 2 such kind of regulatory genes. And we studied the function of all of those regulators in detail. However, today I will only focus on this red labeled PAPR2 regulator here, which belongs to the subtype family of regulators. So when we studied the function of this regulator more in detail, and there I need the next slide. Okay. First of all, we inactivated the gene and studied the pristinamycin production, and there we observed that the inactivation of the PAPR2 gene leads to complete loss of pristinamycin production in comparison to the wild type, which still nicely produced pristinamycin 1 and pristinamycin 2. So therefore, we knew that PAPR2 is an essential activator of pristinamycin biosynthesis. Yeah, can I ask you, are you the one who is moving the slides forward, or can I do that?
No, I don't know.
Okay,
no,
because it's.
I think it's
I'm sorry,
it's a bit left in there.
Yeah, it's really several.
I will ask send me.
But I will. It's okay. I will just move on. So okay. PAPR2 is an essential activator of pristinamycin biosynthesis. This was what I wanted to say. And we studied the function of that regulator also more in detail and figured out where the regulator really binds within the antibiotic gene cluster. So therefore, we purified the PAPA2 regulator and did electrophoretic mobility shift assays, and thereby we could show that PAPA2 really binds to the promoter regions in front of the pristinamycin 1 and pristinamycin 2 structural genes. And we were also able to define a specific consensus sequence for such a binding. And by RT-PCR experiments, which are shown here at the right-hand side, we also could show that there's no transcription of the pristinamycin 1 and pristinamycin 2 structural genes in the PAPA2 mutant in comparison to the wild-type organism. So therefore, we knew that PAPA2 is a direct activator and an essential activator of pristinamycin biosynthesis in Streptomyces pristinus pruritus. And the next slide, which I hope comes in a few seconds, shows you where the regulator is situated within the regulatory signaling cascade. Because it's here located at the very bottom, it directly activates the pristinamycin biosynthesis, and itself it is regulated by a set of other transcriptional regulators where I will not go in detail. However, it's quite complex signaling cascade governed by microbial hormones called gamma-butyronolactones. And what I wanted to mention here is that understanding these kind of regulatory signaling cascades and the regulation mechanisms of antibiotic biosynthesis in general can also help to activate silent gene clusters. And this we observed when we overexpressed the PAPA2 regulator in a foreign strain. And back then, this was Streptomyces lividans. And this lividans strain should come also in a second. This lividans strain usually does not produce any secondary metabolite under standard lab conditions. So this you can see here on the left-hand side, the lividans strain with the anti-vector control. However, when you overexpress the PAPA2 regulator, then the lividans strain produces an intensively red pigmented substance. And this red pigment indeed is undicyl protegiasin. And undicyl protegiasin is a known product from Streptomyces lividans. However, it is usually not produced in the lab, so it's encoded by a kind of a silent gene cluster. And this undicyl protegiasin biosynthesis is known to be under control of a subtype regulator called RED-D. And this RED-D shows quite high sequence homology to our PAPA2 regulator, so up to 55% sequence homology. And therefore, we speculated that PAPA2 takes over the function of RED-D and leads to the activation of this silent undicyl protegiasin biosynthesis in Streptomyces lividans. And we also later on could show that on the molecular level. So I'm really sorry that it's hanging, but yeah, this is what is shown in here. So having that in mind, we had a closer look into this kind of subtype regulator families in general. So in literature, they are described as Streptomyces antibiotic regulatory proteins. They are known to act as pathway-specific activators of antibiotic biosynthesis. And all of them contain a conserved N-terminal DNA binding domain. And with this, they bind to conserved consensus sequences at the promoter DNAs of the promoter regions in front of the target DNAs. However, so far, the regulatory mechanisms, how those regulators really work, is totally unclear. So this is something what we are going to study in the near future. So we will do crystallizations of the regulator and try to figure out how this activation mechanism really works out in the antibiotic producers. So having that in mind, we had a closer look where those subtype regulators occur, and we found that the genes are very abundant in many different actinomycetes. So you can find them in nearly all Streptomycetes, but also in other important antibiotic producers belonging to the actinobacteria family. And when you have a closer look where they occur within the genome, then you can find them as part of biosynthetic gene clusters. And actually, as part of gene clusters that encode for different types of secondary metabolites, like beta-lactam antibiotics, phosphonate antibiotics, polypeptides, and so on. So therefore, we knew, okay, the subgenes are abundant in very different actinomycetes. They are part of gene clusters encoding for different types of secondary metabolites, and therefore we said that those ones are good candidates to be used as general activators of antibiotic biosynthesis. And then we tested this out in a random strain in just a running project of ours, and this also will be shown, I hope, in a few seconds. Because back then, this was a project which we performed with some Indonesian partners. So actually several ones that are sitting in the audience today. So with Puspita Listianti, Vin Kusaryoto, Ira Handayani, Shantaratna Komala, and so on. So back then we aimed to isolate new antibiotics from Indonesian actinomycetes. So yeah, there we are. Back then the Indonesian colleagues isolated up to 422 actinomycetes from Indonesian habitats. A subset of strains was handed over to our labs back then at the University of Tübingen, and we prioritized the strains for the most phylogenetically unique ones. We grew them in several different cultivation media and covered all of that to bioactivity tests to prioritize the one with the best bioactivity. And back then, 9 strains were of most interest, and from these ones we isolated the genomic DNA of, did biosequencing, and then performed a combined genome mining molecular networking approach. And I think this was what Ira mentioned some hours ago in her talk. So this we have done for dereplication purposes and focus on the most unique gene clusters that encode for the potential unique substances. And back then, one of the interesting strains that turned out from this analysis was SHP-227, which showed quite some good activity, and we just randomly used that strain and performed our sub-guided activation strategy with that. So we cloned in our PAPR2 overexpression strain to SHP-227 and got extracts of the cultivation of these strains and tested it against Bacillus subtilis as a test organism, as you can see in here. The SHP-227 PAPR2 overexpression strain showed significantly improved bioactivity against Bacillus subtilis in comparison to the control strains with the anti-vector or the wild-type samples, which means that PAPR2 overexpression led to significantly improved bioactivity of that SHP-227 strain. Now, what you would do in the next step is that you go into the lab and do elaborative isolation of that compound and check what the substance really is. But we didn't do that. In the first place, we started a genome mining approach of that strain in order to find out what might be the substance that encodes for this, or what might be the cluster that encodes for the substance with this improved bioactivity. And as I said, we had the genome sequence in hand. We performed an anti-smash analysis for the prediction of potential antibiotic gene clusters, and thereby we identified 25 potential secondary metabolite gene clusters. And then the question was, which is the one that encodes for this bioactive substance? And then we ran an additional bioinformatic analysis by applying the program PET scan and searched for our PAPR2 consensus sequence. And thereby we found this subtype sequence in front of gene cluster 9. And gene cluster 9 encodes for potential nucleoside antibiotic, which has similarity to a gene cluster from a Streptomyces lineaceus tarapus strain. And that one is known to produce the substances amicetin and plicacetin, which are known compounds. They act as protein synthesis inhibitors with quite good bioactivities. And only when we knew that, then we went into the lab and checked by HPLC analysis what the product of this strain really is. this should be shown in the next slide, hopefully in a few seconds.
Sorry, Yvonne, I'm interrupted. Your time is 3 minutes again.
Okay, I will hurry up.
Sorry.
And I will go through the slides. Okay, so back then we indeed could show that it's amicetin plicacetin that is produced by the PAPA2 overexpression strain and in significantly higher amounts. In the next slide, it is shown that this also helped for the isolation of 4 novel pyrimidine nucleoside antibiotics, which was a collaborative work with the group of Harald Groß at the Pharmaceutical Institute of the University of Tübingen. I'm really sorry, but the slides are just too slow. So this was just published in the beginning of this year. And now we know that this SARP activation strategy really works out. We are now having a set of different overexpression plasmids. I will skip that, but I hope, yeah, I hope that will work. So what we are just doing at the moment is that we apply this to the genome sequenced strains of the DSMC. So we scale that activation strategy up, and we already screened hundreds of genome sequenced DSMC strains for the abundance of SARP genes and SARP consensus sequences. And these are the ones that we are currently using for performing our SARP guided activation strategy. And in one of the next slides, hopefully, I still have the time that also, I will skip that one, that also in our selected DSMC strains, where we clone in our PAPA2 regulator, we observe significantly increased bioactivities in comparison to the control strains. So when we test different test strains as for the bioactivity, and for some candidate strains, we already have some potential substances that are produced. For several strains, you can already see by the eye that something is going on, that when you clone in the PAPA2 construct, that there's significantly production, or that there's production of a pigment in comparison to the wild type strain. And we are currently isolating these substances and trying to find out if there's a novel antibiotic that is produced by those strains. And with this, I'm at the end. I'm really sorry that the slides were kind of delayed, but I would like to thank all the people that have been involved in this project, mainly Ira Handayan, who worked a lot with these Indonesian strains, especially with the SHP-27 strain. Also, Janina Krause performed a lot of these SARP overexpression analysis, and currently the right-hand person are involved in the SARP-guided activation strategy of the DSMC strains. And with that, I would like to thank for your attention. I hope we can move on with joint collaborations, because I really enjoyed that interaction with you. Okay, so thanks, and I would be happy to take questions later on.
Okay. Thank you, Yvonne. Thank you very much for giving such an informative and exciting presentation. Now, let me invite our second speaker, Professor Claudio Tiribelli, MD, PhD.
Here I am.
Okay. Professor Tiribelli. Okay. Okay. Wait a minute. Okay. Okay. Okay, thank you. Professor Tiribelli is the scientific director at the Fondazione Italiano Fegato. He has experience.
Foundation. That's easier.
Oh, okay. Sorry. He has experience as an editor in the Journal of Hepatology and the Annals of Hepatology Journal, and experience as an associate editor in the BBC Research Note Journal. He is interested in research in bilirubin neonatal jaundice, metabolic liver diseases, hepatocellular carcinoma, stem cell in hepatocellular carcinoma, clicker nasal syndrome, and translation medicine. Now allow me to welcome our second speaker, Professor Claudia Tiribili, MD, PhD, to make his presentation. Professor Tiribili, time is yours to give a lecture now. Thank you.
Well, can you hear me?
Yes, of
Can you
course.
hear me?
We can hear you.
Thank you very much for this kind introduction. Let me try to share the screen with you. That's already done. And so I can start my presentation. Let me tell you, it's really a pleasure to be a speaker of the 9th International Symposium on Innovative Bioproduction in Indonesia on biotechnology and bioengineering. What I'm trying to address today, I'm driving the presentation. What I'm trying to do today is actually make a link to what we are eating and drinking and the liver damage. As you know, no, it doesn't work. So why don't we remove my presentation? I'll share my screen.
Okay. Stanley, can you help me?
Yeah, I mean, I need to go on. Let me try to share my screen. And then, here we go. Okay.
So it's moved now?
Yeah.
Okay.
Can you see it?
Yeah,
okay.
I'm seeing it now.
Okay, let's move back to the start. Sorry for that, but okay. So liver is, after the skin, is the largest organ in our body, and in an adult weighs about 1.5 kilo. Although it's not that thick, the normal liver has a volume similar to the soccer ball. And during this period, we are very fan of a soccer ball because it's the largest gland in our body, and 80% is composed by the liver, by the hepatocytes. But most important, the liver is the organ in our body which is largest number of functions. Liver can do more than 600 functions, such homeostatic, metabolic, detoxificant, and immunological function. But in addition to that, and not only a marginal issue, is that the liver is the only organ in our body able to reproduce and regenerate. And this is a Prometeo myth is being depicted. The main causes of chronic liver disease are different things, such as diet, here, medication, viruses, and autoimmune liver disease, moving from normal liver to usually fatty liver, which is increased presence of the fat in the liver, which can go on with the steatohepatitis and then go to liver cirrhosis, which is advanced liver fibrosis and functional complication, ending up with the strip with the presence of hepatocellular carcinoma, which is the end stage liver disease. We need to stop this trip from here to here. And what I'm going to address today mainly is the role of eating and the diet in having liver disease. So, and this is why, because MAFL, the so-called metabolic fatty liver disease, associated metabolic liver disease, has a prevalence globally of more 25% of the population, which means in one out of the 4 European citizens has a fatty liver, so has a possibility to develop a serious liver disease. So if we go from the trip of from normal liver to hepatocellular carcinoma, goes via different steps. First of all, we have the MAFL, the non-alcoholic liver, alcoholic fatty liver disease, which is mainly represented by the increased amount of fat into the liver cell. But once the inflammation occurs, due most probably to oxidative stress, we have a sort of activation of the extracellular matrix of the liver, which is able to create the scares and then move from NASH to cirrhosis, which is labeled by the presence of scars in the liver, making the liver hard and less active. And these scars, together with the regeneration of the liver, end up in the hepatocellular carcinoma, which, as I told you, is the end stage of this trip. We have several risk factors for this. Of course, we have the overweight, the obesity. I'm going to touch this in more details later on. We have type 2 diabetes and insulin resistance. Altogether, overweight or obesity, type 2 diabetes, and insulin resistance make what we know now is called the metabolic syndrome. And the metabolic syndrome is related both in adults, but even more worrisome is present also in children. And this is something we have to think about, because being in children, we need to prevent the fact that our child moves from here to here in the life span he or she is expected. And if we look here, that's very remarkable figure, that if you look the prevalence of obesity from 1975 to 2016, both in females and in males, you may see the dramatic increase in the prevalence of obesity during this year, meaning that the more so-called civilized we are, the more fat we are going to be. And this is something we have to cope with and we have to fight with. And actually, even more worrisome is the trends in the number of children and adolescents with obesity. And if you both in males and in females, this is true all over the world. But if you calculate here, which is the Southeast Asia, also in the Southeast Asia, particularly in the male children are associated with an increased level of fat in the body. And if you look around the world, the presence of the metabolic fatty liver is basically almost the same all over the world. In some regions it's higher, like in US, in South America. In some regions it's lower, as Africa, due to the fact there is a reduction in the calories available. Australia is basically like Europe, but in China we are observing now a booming wave of obesity, and this is something we have to cope with. So going back to what, in my opinion, is the most important aspect of the obesity is the so-called vicious circle of childhood obesity. We have a healthy child which actually due to a wrong life habits, sedentary life, lack of physical activity, is getting mildly obese child. And the mildly obese is actually a child is less prone to exercise, and those goes from mild to moderately obese child. And if this is also keep going with a wrong diet, is going to move from moderately obese child to severely obese child, and actually to and then to obese adult. And this is where we are going to go from a healthy child to a potentially sick child with the presence of fatty liver. And if you look around, you see that genetics may play a role in obesity. And we know now that the contribution of heredity to the risk of obesity is 25%. And this variability is related to the genetic background of the different studies performed. So we need to understand that habits, in addition to genetics, is the main predisposing factors, particularly habits of parents living with the child. And we just published an editorial in an international peer-reviewed journal, was APT, making clear that it's the parents giving us the wrong information how to behave, less exercise and much more sedentary life. What are the targets of fat accumulation in the body? Of course, obesity is a multi-organ and multifactorial disorder. We can have that damage in the liver, in the brain, in the heart, in the vessels, in the nervous system, and in the pancreas, and in the muscles. All together, meaning that we have a lot of complications of obesity in the childhood. We have damages in the behavior, psychosocial, neurological, like say the tumor. We have pulmonary, sleep apnea, asthma, and intolerance of the exercise. Of course, cardiovascular, due to the dyslipidemia, hypertension, coagulopathy, gastrointestinal, as I told you about steatohepatitis, and also renal, endocrine, and muscle. So this, I mean, we have to conclude that an obese children, like an obese adult, are sick, and we need to cope with. So which are the main causes? Is the imbalance between the calorie taken and the calorie expended. I mean, I'm used to say that the weight is like our bank account. If you eat, I mean, if you gain a lot of money, you don't spend, you get rich. But if you eat a lot of calories, but you spend a lot, you get poor. So we need to understand that globally, there's a striking decrease in physical activity. 1 in 5 boys and 1 in 10 girls is not having a real and appropriate exercise. And even more worrisome is that the amount of fruit eaten by our children is drastically reduced as compared to the ideal amount. Only 30% of boys or roughly 40% of girls eat a fruit a day, in spite of the fact that fruit is readily available all over the world. And the major suspect is sugar. Sugar, the white sugar, it's really being associated with the presence not only of obesity, but also liver disease. And the question is how much is too much? And we need to understand this because we can then give advertising. And here is what we have. And what we have here in the slide is coming from the World Health Organization, telling us the content of teaspoons for each beverage. And mind that the WHO recommends consuming no more than 12 teaspoons per day. If you drink a Coca-Cola, you drink it's like you eat 8.5 teaspoons of sugars. If you eat a yogurt drink, it's like 7. But if you need an energy drink, you have 10. And of course, telling you that what we are using to give our children, the so-called sodas, are then maybe very dangerous. And here's what we have in as comparison. Of course, the blank is the water. There's no sugar. But if you look at Sprite and Coke, which is widely used, we have that 33.7 and 35 grams of sugar per each can. And even worse are Fanta and Schweppes, when we go from 33 to 43 and 44 gram of sugar. So telling us that, I mean, we are not surrounded by a friendly beverage consumption. What is the total fat? The total fat should not exceed 30% of the total energy intake. And we have different kinds of fat. We have trans fats, the dangerous one, due to the so-called junk food. We have the saturated fat, which is related to coconut, palm oil, and alimentary, polyunsaturated skin, meat, and fruit. And we have then the unsaturated fat, which are the real good fat, that should be much higher than 90% of the total fat in intake we have every day. And this is important to prevent additional damage. How the excess of fat?
Sorry, Professor Tribuli, I may interrupt you because your time is only 4 minutes again.
Well, my time is still 5 minutes.
Oh, 5 minutes. Okay, 5 minutes again.
I mean, according to my watch, it's still 5 minutes.
Okay.
So I'll be on time. Don't worry.
Yeah.
So how the excess of fat and sugar impact the liver? This is a cartoon. It's a very complicated cartoon. I've had no time, unfortunately, to go through. But the key issue is that insulin resistance and increased intake for the diet bring together the possibility to accumulate fat in our liver. And this is what we have to deal with. So do we have animal models to study the juvenile fatty liver disease? And the answer is yes. This is work we have done in our lab, driven by Sylvia and Natalia and Pablo. And what they did, they reproduced the fatty liver, a juvenile fatty liver in a mouse, given the mouse a lot of fat and a lot of calories. And what we have, what they have been able to reproduce is a fully scaled presence of fibrosis. Mimi telling that this may be a good model to deal with in order to understand if there is any good treatment to work with this. In addition to that, we know that fatty liver is really linked to a leaky gut, because the gut is living alone with a lot of toxins moving from the gut to the liver. And this is what we have observed, that in large intestine, we have a leaky, the presence of a less tight junction as compared to controls. So in conclusion, we can say that excessive sugar and fat since young age induce obesity-related multifactorial events that lead to progressive multi-organ dysfunction, fibrosis, leaky gut, altered glucose homeostasis, means diabetes. And this is, and we have to realize that the progression in children is much faster than adults. So we need to take care of this aspect for the future generations. And we know now that in the future 20 years, all the liver transplantation will be related to fatty children at the moment. So we need to take action on that. So let me skip all this because I have no time here. Let's try to deal with the future perspective. We heard that insulin resistant type 2 diabetes and obesity is the accounts for metabolic syndrome, which is a booming of obesity. And today we are still missing something, but we need to do something as a diagnosis, screening, and efficient treatment. And the clock is still ticking. So thank you for attention. I think I did my job in time,
Okay,
and I used all my 20 minutes.
Okay, thank you very much, Professor Tribili. Thank you for giving us such an informative and interesting presentation. And ladies and gentlemen, now are the time for the question and answer session. We have 20 minutes for Q&A session. If you have question for both speaker, please raise your hand and or you can write the question in the chat room. Okay, I think we have wait a minute. I need to see. I think Miss Atika, are you ready to give your question?
Thank you, Dr. Yeah, my question is to Dr. Yvonne. She's gone. No. Yeah.
Okay.
So,
Yvonne, Yvonne, are you still in here?
I'm still here. I'm
here.
here. Yeah. All right. Okay. So my question is a bit practical now, but I never done the analysis that like what you did. So my question is like how long actually to get from your analysis of biosynthetic gene cluster until the analysis of the mutant, if I'm not mistaken. It's like you did like cloning for all this experiment, right? If I'm not mistaken. So ideally, how long until you get the results? Thank you.
Yeah, thanks for the question. That's actually hard to say because it's mainly dependent on the strains to manipulate. Actinomycetes are not so easy to genetically manipulate, and depending on which kind of organism you have in hands, this really depends how fast you are. But concerning the whole progress or process, depending if you have the genome sequence, this is the first step. Let's say if you have the genome sequence, then you could go for the anti-SNP analysis, and this is quite fast because you just upload the sequence on the anti-SNP tool, and then you directly get the output after a few minutes. But this is, let's say, pure bioinformatics. It can give you an estimation what kind of gene clusters may be encoded, but you then need to really have a closer look into that and study that more in detail and also manually. And then I mean, nowadays we have these overexpression plasmids. In hence, they are cloned. Then you just need to introduce them into the actinomycetes. And if everything works well, you are maybe successful after 1 or 2 weeks. If it's a strain that is harder to treat, then it can take several months, I would say. But this is what where I skipped a slide, because what I wanted to mention is that nowadays we have different genetic tools. So we have different plasmids, which can be used either for protoplast transformation, which is a process how you can get DNA into the strains, or you can use the plasmids for conjugation, where you use the opportunity that you have an E. coli strain that can deliver the plasmid to the actinomycetes.
Okay, Fawn, thank you for the answer. And in the chat room, I have 3 questions for Professor Tribeli. First question is, it is true that some herb like curcuma is help in maintain our liver health? Thank you. This is from Mr. Elok. Miss Elok, I think. Are you ready to answer?
Yeah, the answer is maybe. I mean, there's no real confirmation that any drug is preventing liver disease. There are some drugs used to or suggested to be helpful, but I mean the real scientific demonstration is not yet. The curcuma is one. The other one is silymarin, the thistle syrup. Others are the extract from the apple. But I mean, there are some information, but not real specific and solid scientific demonstration. So we need to take this with a very, with caution.
Okay, thank you for the answer. Okay,
Second
and the second question is, what are the most recent and reliable treatment to deal with liver damage that are already on cirrhosis stage? Is surgery and transplantation are the only option?
Well, it depends about the stage of the disease. Of course, if you have an end-stage liver disease, transplantation may be an option. Actually, it is an option. We have to realize that in Europe, every year we are performing roughly 2,500 liver transplantation all over Europe. But I mean, the ideal is not to go to the liver transplantation. The ideal issue is to stop the trip before moving ahead. This is what I try to demonstrate in my presentation as far as obesity is concerned. If we start changing our lifestyles by asking our adolescents and ourselves to exercise more, to eat less, and to be much more pragmatic in terms of daily behavior, this could be very helpful. Not only for the liver, but also for cardiovascular disease, for diabetes, for everything.
Okay, thank you for the answer. And next, are there any possibility to recover from any damage in the liver?
For what? For?
Are there any possibility to recover from any damage in the liver?
Absolutely. I mean, liver is a regenerating organ. If you have a acute hepatitis, I mean, and you heal from the hepatitis, I mean, like A hepatitis, the liver returns back totally to normal structure. So we don't have to have any scar. Of course, if you don't recover fully, for instance, from B hepatitis or C hepatitis or other diseases, then the situation is not acute, it's going to be chronic. So the damage is chronic. So the end result is that the liver is daily damaged by the heating stimulus, and this ends up with something which is chronic liver disease.
Okay, thank you for the answer. And now we still have 13 minutes left. If there are any question for both of speaker?
We have been too clear.
Okay.
Or too confused.
Okay.
I have no idea.
If there any, if there's no any question again, I will ask maybe to Professor. Okay, Professor Mas. I want to ask Professor Iwan, what medium was used for self-activation with genome sequence in DSMZ strand? Is there any effect of the medium on the successful use of the self strategy in activating the silent gene cluster?
Yeah, actually, also here we tested a different set of cultivation media just to make sure that we do not miss anything. So actually, we tested 3 to 5 different cultivation media. Some of them you may know. I will not go in detail with these ones, but it turns out that if you have activation, that 1 or 2 cultivation media are enough. So it looks like that really the abundance of the subtype activator is more relevant than the media you use.
Oh, I see. Okay,
Okay,
thank you, Yvonne. Yeah,
but Ira, I think I also see a question in the discussion panel or in the chat room
I will check
from Hatati. I may also read it by myself because the question was what kind of data that one inputs to get biosynthetic gene cluster using Plantis mesh. So I'm not sure what if this is if I understand the question correctly, because there are different tools available. So AntiSmash is the program that can predict antibiotic biosynthetic gene clusters encoded in bacteria, and PlantisMesh is a tool where you can identify gene clusters from plants. So if this question really targets PlantisMesh, and the question is which kind of biosynthetic gene cluster one can find there, I would say that you get an output which mainly contains gene clusters encoding for terpene substances, type 3 polyketide gene clusters, alkaloids, and so on, because these are more substances or gene clusters that are more abundant in plants. In bacteria, you may get just another output also on gene clusters encoding for non-ribosomal peptides, type 1, type 2 polyketides, for example. So I hope I could answer that question.
Okay. Yeah, thank you, Yvonne. If there are any questions again for the bot speaker, please raise your hand or write it in chat box. Maybe I will ask Professor Claudia Terribili. Does consuming too much fiber also causes liver damage?
Yes. I mean, the point is not what you consume. The point is what the amount you consume and the calories. I mean, if you eat, let's say, a kilo of bread, the amount of calories is much higher than a kilo of salad. But of course, the taste of 1 kilo of bread is much better than the taste of 1 kilo of salad. And if you put some butter on your bread, the calories increase. So I think we need to make a balance of our daily behavior. I mean, if we eat more calories, we need to spend more calories. As I told you during my presentation, our weight and our metabolic functions is like the bank account. I mean, if you gain a little amount of money, but you are so able not to spend, you're getting rich. But if you gain a lot of money, but you spend a lot, you spend more, you get poor. So everything is balanced. We need to be careful. And then the basic point is to teach our children to behave properly. And our children, at least in Europe, don't behave properly. They are in front of a screen. They don't move. They don't go to walk. They don't go to play football. They don't go on the bike. They stay home, and this is bad. That's why we're having a real problem with it with adolescents.
Okay. My next question is there any effect of a supplement on the liver health?
You mean the supplements?
Yeah, like vitamin?
Well, I mean, the supplements have never been demonstrated to be helpful. The supplements are very helpful for the companies that are making the supplements. they're making a lot of money they're making a lot of good for the producers of the supplements, but there is no evidence that they're making good for the liver of the subjects taking the supplement.
Okay, thank
So
you.
take a message, don't take supplements.
Oh,
Eat the
okay,
fruit.
thank you for your
Eat
suggestion.
the fruit, it's more than enough.
Oh, I see.
You're
Okay.
lucky enough to live in a country like Indonesia, when the fruit is so beautiful, and you have mangoes, you have everything.
Yeah.
Why don't you have to take supplements? If you're living in Europe, I mean, you go and then eat an apple or an orange or a pear, and that's it. It's better, it's cheaper, it's much more healthy. Trust me.
Yes, of course. Thank you for your suggestion. Okay, if our time is left 2 minutes. Okay, or Yvonne, do you want to ask the question to Professor Tribelli?
Yeah, actually, I would also want to ask a short question to Mr. Tiribelli. I wondered if there are any numbers. What is in general more harmful if one drinks too much alcohol, or using too much calories from early days on?
I didn't touch alcohol for obvious reasons, because alcohol, there is a safe amount of alcohol daily, which around 30 grams per day, both in females and males. 30 grams of alcohol per day means half a liter of wine, or a small glass of super alcoholic schnapps or whatever it is, which means that if you have one glass of wine with your meal, it's good. What is bad is to drink outside the meal, because the absorption of the alcohol without any aliment is very quick. This is why the heavy drinkers are used to eat together with alcohol something which is oily, like olives and peanuts, because you have a sort of film of oil in your stomach, so the absorption is less, and then you can drink more. That's the trick they're doing. So a limited amount of alcohol is safe, actually, but it depends on how limited is limited. And a good glass of wine is good. And let me tell you that, unfortunately, I'm a teetotal. I cannot drink alcohol, not because I don't want, because I cannot stand it. And this is my limitation, because with a good meal, a glass of wine, assuming that your religion is allowing to do it, it's good.
Okay, thank you for the answer. There's 3 questions again in the chat room. First, for Professor Tribelli. The question is, as we are getting older, is it true that our metabolic rate are also slower than when we are young?
Yes. Yes, the answer is yes.
Yes, okay.
That's it.
Yeah. And there's 2 questions for Yvonne. Did you also conduct any research in Germany on the culture to activate gene cluster for the production of natural products? Based on your experience, which substances from terrestrial and marine ecosystem produce more new metabolites at this time?
Okay, concerning question 1, we didn't do co-cultivation approaches, and I tell you why. Because later on, the identification of the produced metabolites is just too hard. If you do co-cultivation, you may activate something, but you do not know what is produced, and then you need to go again into the lab, isolate the substance, do chemical analysis to really identify the produced substance. We had the attempt to do a targeted activation, because if we use these kind of regulators, then we exactly know where they bind, which kind of antibiotic gene cluster is activated, and this facilitates later on the identification of the compound. And therefore, we didn't do co-cultivation, which also may work, but we only focus on this targeted activation approach. So the second question is, which kind of organisms do produce more antibiotics, terrestrial actinomycetes or the marine-derived ones? We cannot say until now. you can have marine organisms with really a lot of antibiotic gene clusters, but also terrestrial ones can harbor more than 50. So I don't see a difference in terms of the potency of terrestrial against marine organisms. So there's still enough out there to be identified in terms of new secondary metabolites, I would say. Okay, but I think there's an additional question. Ira, would you like to?
Okay, I think there is no any question again. Okay, I think time is finished. Time is the end for the question. Okay. Finally, we come to the end of this seminar. I want to draw a conclusion from what speaker said. From Professor Ivan Mas, we can learn that a self-guide strategy can be used to activate antibiotic biosynthetic gene cluster in a targeted manner and thereby exploit the biosynthetic potential of actinomycetes. And from Professor Tribuli lecture, we can conclude that excessive sugar and fat consumption from a young age can lead to the obesity, leading to the development of metabolic associated fatty liver diseases and non-alcoholic fatty liver diseases in the children. Unfortunately, the development of metabolic associated fatty liver diseases in the children is faster than in adults, and efficient therapy to treat this disease doesn't yet exist. Hence, the best options still depend on the lifestyle changes. Okay, finally, I would like to thank to Professor Ivan Mas and also Professor Claudia Tribuli for their informative and interesting presentation, and all the participants for their very active participation. And now I will hand over the event to the MC. Thank you. Miss Aulia, time is yours now. Thank you.
Terima kasih.
Terima kasih banyak.
Thank you, Professor Tribuli, Professor Ivan Mas, and also Dr. Ira for the amazing and also presentations this afternoon. Distinguished guests, ladies and gentlemen, now we have arrived to the end of today's conference. On behalf of official committee, I would like to thank you for your attendance and also active participations on the first day of ISIBio 2022. We're signing off. See you on the second day of 9th ISIBio tomorrow at 9 AM Jakarta time with another interesting speakers and also materials. Stay safe and also healthy.
Bye-bye. Thank you.
Thank you very much, Yvonne and Professor Tribuli.
Thank you. Bye.
Bye.
Bye-bye.
Bye-bye.